Hello again. Thanks for joining me, Dr. Arpin, for your second pre-lab video. In this video, we'll be going over the assay portion of our awesome experiment, hemoglobin drug synthesis and assessment. So don't forget, our whole goal here is to identify drugs that bind to hemoglobin, and you're going to identify those drugs by preparing them and testing them yourselves. And so by doing this experiment, I hope you gain exposure to MedChem. You're going to execute a very cool protein binding assay, and you're going to analyze your own as well as others' collected data. Remember also that there were three main parts of the experiment: the synthesis, assay, and analysis. You completed the synthesis, most of you hopefully on day one, and so now our focus today is on that protein binding assay. So of course we should review how this assay works again. Remember that we are looking at the hemoglobin protein, which is actually a tetrameric protein. And here's a monomeric portion of hemoglobin with the heme right there in the middle. It's that green molecule. So what we've done though is we have treated this with acidic butanone to actually extract that heme portion of the protein. And that resulted in our isolation of apohemoglobin, which is just hemoglobin with an empty active site. And so to assess how well your drug actually binds to that empty active site, you're first going to take the highly fluorescent complex of the apohemoglobin protein with that small molecule ANS. If you recall from our first pre-lab video, when these two are in solution, they form a complex that is highly fluorescent, but that binding of ANS to the apohemoglobin is reversible, right? And it can be displaced. ANS can be kicked out. So that's exactly what we're going to do with our drug. And if we follow this via the fluorescent signal, we should get a nice dose response curve, as shown here, that first has a really strong fluorescent signal corresponding to that ANS-AHB complex. But then as we introduce more of our drug, then that ANS compound will get displaced, will get kicked out, and we will see the loss of fluorescence. And then after obtaining that dose response curve, we'll be able to identify the IC50 value, or the concentration at which we see 50% inhibition, or in our case displacement or binding to that AHB protein. And this is the measure of which we use to compare the different drugs. And so as we see here, we have three different drugs, and the drug with the lowest IC50 is drug A, and that means that it is the most effective or the most potent drug, in our case the best binder, because we need the smallest amount of drug A to get that 50% inhibition or 50% binding. So we need the smallest concentration. That makes it the most effective drug. So we're going to collect our dose response curves, we're going to identify our IC50, and then we're going to compare it with the other drugs that everyone made and compare their IC50s to identify those best binders. Alright, so to carry out our protein binding assay, the procedure can be summarized in just 4 general steps. First, we have to make a stock solution of our drug. And using this, we're going to perform a serial dilution to get that drug then at different concentrations. Then we're going to make that fluorescent AHB ANS complex. We're going to make a 5 micromolar solution by simply combining the AHB and ANS in a 1-to-1 ratio. We're then going to combine the drug with that fluorescent complex. We're going to have the drug at different concentrations and just mix it with the complex. And then finally, you'll get to collect the fluorescent signal of each of those different concentrations with the complex. All right, step 1, drug preparation. First thing we're going to do is make that drug stock solution. We do that by adding about 25 milligrams or 0.025 grams of your drug into a 1 gram vial. It looks like this. Okay, you want to label this drug 30 millimolar. It's the first thing you want to do. Labeling is very important throughout this experiment because we'll be using lots of different vials. So you want to weigh out about 25 milligrams of your drug into this vial, and this is easy to do if it's a solid, or you can just dump it in. But if your drug is an oil, it may be easiest to use a glass pipette to suck it up if it's a thin oil. But if it's really sticky, you might need to use a small spatula. Just somehow get about 25 milligrams of your drug into that vial. And whatever mass you do get into the vial, you'll then want to input that value on your Excel sheet. Okay, so this is provided on Blackboard. There are 2 tabs in that Excel sheet. If you look at the bottom, and we're in the solution prep tab. Okay, so as soon as you have weighed out your compound, make sure to input all of the relevant information here. Okay, you need to definitely put in the molecular weight of your drug. Okay, and you want to put in the compound name. You can give it your name or the name of the drug in the handout. But whatever mass you make into that vial, just put it here, and then you'll see that it populates with an amount of DMSO to add to that drug. So then next you're going to want to actually pipette that amount using a micropipette into your 30 millimolar 1 gram vial. Okay, so you got your drug, and now you want to pipette in that amount of DMSO. So if you've never used a micropipette, you probably want to check out this video. It has some really nice tutorial on the technique, or you can ask your instructor about how to use a micropipette. After you've done that, you want to obtain a scintillation vial. So these are different. They're bigger than the 1 gram vials, and they have white caps. But you want to obtain one of these vials and then label it drug vial 1. And then into this vial, you'll want to add 900 microliters of DMSO. Do this with the micropipette. Then get out your 30 millimolar solution and add 100 microliters of this solution to your drug vial number 1. Okay? And then the last step is to add 9,000 microliters or 9 mils of phosphate buffer. So the buffer will be provided for you, and this keeps everything nice and stable in solution. And so this is what we're doing here is a dilution. We started with 30 millimolar, but now after adding all of this DMSO and phosphate buffer, you now have a 300 micromolar solution of your drug in 10% DMSO. Nice job. Alright, so now we have our drug stock solution, but we need to get the drug into different concentrations so we can test it against the complex. Okay, so we're going to get them in the different concentrations by performing a serial dilution. To do this, first obtain 7 1 gram vials, just pick them up, and then label them drug vial 2 through drug vial 8. Label each one. Again, labeling is very important because we have so many vials. Go ahead and place these in a line on your pegboard, and then go ahead and pipette in 1,000 microliters or 1 mil of the phosphate buffer into each vial 2 through 8. Okay, not this stock solution, just drug vial 2 through 8. Do this using a micropipette. So we're now ready to perform the serial dilution part. Okay? Take 1,000 microliters from drug vial 1 and micropipette it in to drug vial 2. Okay? This already has 1,000 microliters of buffer, so by doubling the total volume, we've now performed a 2-fold dilution. Okay? So we've diluted our original drug now. Do this and mix the solution by just simply taking it up and forcing it out of the micropipette a few times, but then you just repeat this process. Okay? Take 1,000 microliters from drug vial 2, add it to drug vial 3, take 1,000 microliters from drug vial 3, add it to drug vial 4, and so forth. Okay, and each time we are getting more dilute because we're adding it to more buffer. Okay, so we're doing a 2-fold dilution each time. So then at the end, you will have 8 different concentrations of your drug, each one becoming more dilute until you get to drug vial 8. Again, nice job. So importantly, you still need at least a half an hour to finish out the assay. And that's assuming that the fluorimeter is free and you can jump right on the instrument. So if you don't have a half an hour left in the lab period, I would just call it a day and pick up here next time. But assuming you do have a half an hour left, your next step is to prepare that brightly fluorescent protein ANS complex, the apohemoglobin ANS complex. So you do this in another scintillation vial, label it AHB ANS complex, and you want to secure that vial in an ice bath. So just clamp it to your bench and then put a little cup of ice around it. And this is to keep the protein nice and stable, to keep it from degrading. So again, this is why we need at least a half an hour left, because we don't want that protein to degrade. So after you have that vial in an ice bath, okay, the next thing you got to do is input the provided AHB absorbance. So this will just be written on the board. My student will collect it the morning of, and this corresponds to the concentration of the protein in solution at that time. Okay, so you want to input that absorbance right here. This is part of the HB Drugs Excel sheet in the Solution Prep tab. So you're going to put that absorbance there, and what's going to come up then are the amounts of the protein solution and the amounts of the ANS solution that you need to add to that vial. Okay, so you can see here whatever that absorbance is, you'll then be given how much protein solution to add, okay? And then you'll also be given how much ANS solution to add. So you just add both of those to your vial using the micropipette. And then you want to add buffer in order to make a 5 micromolar solution. So all of that's been provided right on the Excel sheet. And after you add all of those to the scintillation vial, you have made a 5 micromolar solution of a 1-to-1 complex of the apohemoglobin ANS. Alright, so we've finished steps 1 and 2. We've prepared our drug as well as our complex. So the next step is to combine the two. Do your best to place your pegboard into a large ice bath. I recommend getting one of those big styrofoam containers, putting some ice and water in it, and doing your best to get that pegboard right in that ice bath. You'll also want to put your AHB-ANS complex file in there. This will keep the protein cold and keep it from degrading. Again, this is why we need to finish the assay today, because we want to keep our protein from degrading. Next, you'll want to obtain 9 microcentrifuge tubes that look like this, okay, and put them in your pegboard, all in a line. Okay? You'll also want to label them number 0 through number 8. Okay? And then into each microcentrifuge tube, you want to micropipette in 1200 microliters or 1.2 milliliters of your AHB-ANS complex. Do this into each of your tubes, number 0 through number 8. Next, just into tube 0, you want to micropipette in 400 microliters of just buffer. Okay, this will serve as our protein with no drug. Okay, we're going to see how much signal we get just from the protein complex. But into the remaining tubes, you'll want to micropipette in 400 microliters of each drug vial into the corresponding microcentrifuge tube. Okay, so from drug vial 1, you want to pipette in 400 microliters into your tube number 1. Take 400 microliters of drug vial 2, add it to tube 2. Take 400 microliters of drug vial 3, add it to tube 3, and so forth for all of the remaining vials. So you've now mixed the drug and your complex, and you'll want to cap all of your tubes, and you'll want to invert them gently just a few times and let them sit and reach equilibrium. But then after that, you will have 8 different concentrations of your drug mixed with your complex. Congratulations, you've made it to step 4. You are now ready to collect the fluorescent signal from your samples. You first need to obtain 9 fluoromina cuvettes, and you want to place them all in a line on your pegboard. Okay, and try and keep them all in a straight line from 0 to 8. Okay. Then you simply want to transfer all of the contents from each microcentrifuge tube into its corresponding fluoromina cuvette. Okay. Importantly, you need to use a new glass pipette for each sample. We don't want to see any contamination here. So with your next tube, just use a new glass pipette and transfer all of the contents from the microcentrifuge tube to the fluoromina cuvette. Okay? Just repeat this for all of your samples, and it's very important that you know exactly which cuvette has which sample. Okay? You can't label the cuvettes, but you need to keep them in the right order. Okay? So you need to make sure you know exactly which cuvette has which sample. You're now ready to go to the fluorimeter, so take your large ice bath with all your cuvettes and your notebook down to the fluorimeter in 318. When you walk up to the instrument, you'll see the parameters window on the right hand side. You need to make sure that it's still set up correctly. So the excitation wavelength needs to be set to 365 and the emission wavelength needs to be set to 400. These excitation slits and emission slits should be at 10, but most importantly it should say ready in the bottom right corner. So if this is all set up, you can go ahead and insert your first sample, cuvette 0, your sample 0, place it right into the instrument. You do have to open the door to the instrument to access the sample holder, and then you just insert your cuvette directly into the sample holder. You will then close the door on the instrument, and next you need to hit the measure icon. Okay, it'll be on the right hand side. Just click on measure. It'll take about a minute to collect the scan. Once the scan is done, a window with your data will show up in the middle of the screen. If you can't see the top of the peak, just right click in the middle of this window and click auto scale. Okay, but you should get a spectrum like this with a nice peak. And so what you need to do is take the line in the middle of the window and drag it, click on it and drag it to 455 nanometers. And you can see, you can check to make sure you are exactly at 455 by reading this number in the top corner. So again, you want to drag it to 455, and then here is your fluorescent signal. So this is what you want to write down in your notebook for this sample. Okay. Once you've done that, it's important that you then minimize the window, because this will bring you back up to that measure icon so you can take your next sample. And then you just put your cuvette right back into your pegboard and move on to the next one and repeat this measurement process for each of your cuvettes. Make sure you've written down the signal for each cuvette and the correct cuvette each time. Then it'll be easy to transcribe right into your HB drugs Excel sheet, all that data, and you'll get a nice curve. And then you're all done. Make sure to let the next person in line know that they can jump on the instrument if there's anybody waiting. And the next portion is the analysis. Hooray!